All right. So, hello everyone. Welcome back. So, I need to speak louder. So, yeah, this time we only have one microphone. Still debugging here. So, yeah, welcome back. We will start with the second session of this morning before lunch. And we will start with Thomas from Evangelidis, who will give us these questions.
Thank you. Thank you for the introduction. Can you hear me? Yes. All right. Good morning, everyone. Welcome to Prague. I am Thomas Evangelidis. I am the founder, CEO, and CSO of the company Evangelidis, which is based in the Czech Republic. We have the experimental facilities in Brno and the computational facilities in Prague. And I'm going to present you our technology, which harnesses the powers of NMR and AI to enhance drug discovery. So I will skip the introductions because you're all familiar with cheminformatics. So what we develop is an integrated NMR AI platform to address the three stages of early drug discovery, starting from the structure determination. We use primarily three-dimensional, four-dimensional NMR experiments, and we have developed the software, which is called 4D-Graph ID. It's almost ready for commercialization. So following the structure determination and the study of the dynamics, we proceed with virtual screening and hit discovery, for which we have another deep learning based software. It's called Deep Hit Miner. It's also almost ready for commercialization too. And it uses primarily one-dimensional NMR data as input. And finally, the last stage we address is the hit to lead, when we try to optimize the structure of the hit compounds. And for that, we have another software. It's called Deep Scaffold. And that one utilizes primarily one-dimensional and two-dimensional NMR data as input. So what sets us apart from the rest of the chemoinformatics companies is that we employ ultra-fast NMR experiments to study the ligand interactions at the atomic level, both from the ligand's perspective, but also from the protein's perspective. And here in the slide, I show you two examples of spectra. At the top right, you see a two-dimensional spectra, which gives the amide peaks of the protein atoms, primarily of the backbone. And specifically, you see an overlay of three two-dimensional spectra that correspond to gradually increasing ligand concentration. So we gradually increase the ligand concentration with that trait to see which peaks they start to be perturbed, as we say. And I have color with green color, green circles. Those peaks that have changed location, they are perturbed, and they correspond to the amide residues, amide groups that interact with the ligand. However, this protein is peculiar, it's intrinsically disordered, and you could see some new peaks appearing in the spectrum, which I colored with cyan color, the light blue. And they appear at high ligand concentrations, which means that the ligand somehow stabilizes some of the flexible residues in the loops. And they fall within the timescales of NMR, so they start to appear in the spectrum. So this is very valuable information. At the bottom, and we can collect dozens of such spectra per day using autoassemblers like the one I showed in this figure here. So the other type of input we use is ligand-observed one-dimensional spectra, which you see at the bottom. In such kind of spectra, we see only the atoms of the ligand. For example, here I show you a small fragment and the respective spectra we have obtained for the proton. With blue, you see the reference spectrum, which is the spectrum of the fragment alone in the solution. And with red, you see the spectrum of the mixture of the protein in the fragment. And from the changes, we can guess which atoms of the small molecule interact with the protein. And this is the so-called ligand epitope. It's very important information, which is utilized by our deep learning algorithms. So overall, our NMR AI platform fuses these two types of data to discover new hit compounds and grow them or add fragments, and proceed with iterations of design optimization. So in this workflow, I show you how we address the hit discovery. As already mentioned, the input are ligand-observed one-dimensional NMR spectra. Yeah, like this one. And from this, we can extract the atoms of the ligand epitope that interact with the protein. So the chemical structures, along with the ligand epitope, are then the input to the neural network, which we train and virtually screen large chemical libraries to score the compounds. Following that, we have developed a multi-objective genetic algorithm that can select a diverse set of compounds, which are chemically diverse. They have high score, binding score, assigned by the neural network. And they can also have another or two optimized properties like the solubility, for example, which is commonly used. And the final stage is, of course, the visualization. Here it can be seen better. We project the chemical library to 3 UMAP dimensions. Each sphere represents one atom. And actually, the size of the sphere is proportional to the score assigned by the network. You can also display other properties, like in this example, the color represents the solubility. But we have a long list of properties you can select in the graphical user interface. And when the medicinal chemist does the selection of the molecules for the next screening round and clicks on each of the sphere, can display the chemical structure at the right. Now, the pointer is lousy completely, so I will use the mouse. So as you see here, apart from the chemical structure, you can visualize the predicted atoms that interact, like predicted ligand epitope, we call it. So, and actually, the network assigns a probability to each of these atoms that is proportional to the strength of the interaction that mediates. For example, this aromatic proton has higher score than the other 2 aromatic protons of the ligand epitope. So this is in a nutshell how we do the selection of the compounds for the next screening round. And what we're currently working on is fusing 2 types of networks. One that takes as input the ligand observed NMR spectra, namely we see from the perspective of the ligand, the interactions, and the protein observed NMR spectra, where we see from the perspective of the protein, the interactions. Right? So it fuses these 2 networks to predict the final binding probability, but also the atoms of the ligand epitope and the atoms of the protein that are predicted to interact with the new molecules, the new unseen molecules in the libraries. All right. So besides the drug discovery software, we develop software for protein structure determination, which is based on higher dimensional NMR spectra. Here in this slide, I showed you an example of 3 classes of proteins. At the right, you see a structured protein whose 3-dimensional structure we can solve by X-ray, cryo-EM, NMR, even AlphaFold. And the other AI-based algorithms can predict pretty well the 3-dimensional structure of folded proteins. Welcome in a single domain. But in the second class, where you see the partially disordered proteins, the only biophysical method suitable to study their structure and dynamics is NMR. That's the reality. And at the bottom, you see 2-dimensional spectra. The first dimensional spectrum from the structured protein is well resolved. The peaks are nice, and you can separate them. In the second case of the partially disordered protein, you see that the peaks, they start to overlap some regions in the center. This is called spectral crowding. It's very typical for disordered proteins and becomes even more severe for the intrinsically disordered proteins that are completely disordered, in which case the spectral crowding is so severe that 2-dimensional spectra are completely unsuitable for the study of the dynamics of the protein. So what we do in this case, we add extra dimensions. Every time you add an extra dimension, you separate the peaks, right? So usually for large IDPs, not even 3 dimensions are sufficient. That's why you have to add 4 dimensions or 5 dimensions. So our solution is based on that. That's a unique selling proposition. And it's ideally suitable not only for IDPs, but also for large proteins above 20 kilodalton, because as some of you may know, the size limit of NMR is around 20 to 25 kilodalton. The more atoms we have in the protein, the more peaks you have in the spectrum, and therefore the more overlap. So that's the strength of our solution. When we do NMR of proteins, we usually have 2 options. The first option is to study only the backbone of the protein, which you see at the left. And for that, we typically require to measure 5 3-dimensional spectra. That's approximately 1 week of measurement. However, if we want to study the full 3D structure along with its dynamics, Then with our method, we have to measure additionally 2 4-dimensional spectra. And that translates to another 3 weeks of measurement on average. And here I show you a comparison of our software 4D graphs on backbone assignments with best available software for the same purpose. It's called FLY, it's from NMRTIS, the FLY algorithm. As you can see here on 88 proteins, most of the proteins our 4D graphs outperforms. It can predict, it can assign more correct resonances to atoms. That's the value you see at the vertical axis, percent correct assigned. The median is 97% versus 93%, something like that. So we have superior performance on that task. And why is that important? That is very valuable for intrinsically disordered proteins like the androgen receptor, whose metadynamics ensemble you see at the right. These proteins are completely unstructured, particularly this protein, which happens to be our target for drug discovery, has only a small stretch of residues, about 70 residues, that assume some tertiary structure, 3D structure, although, as you can see, they are unstable. So applying structure-based approaches on such proteins is futile, because when the ligand binds, it reweights the whole ensemble unpredictably. So what is most important here is to map the atom-level contacts, and specifically the contacts between the atoms of the ligand and the atoms of the protein. And this is exactly what we do with our NMR-based technology. We see from both ligand's perspective and protein's perspective the interactions. That's why it's ideal for intrinsically disordered proteins like this. So how it works, as I already mentioned, if we want only backbone, we measure 3-dimensional spectra. If we want also side chains and 3D structure, we measure another 4-dimensional spectra. And 4D graph ID does the automatic resonance assignment, namely associate resonances in the spectrum to atoms of the protein. And then extracts the restraints, distance restraints, backbone torsion angle restraints, and provides them to a modified version of OpenFold, which has been modified to use both the AI components along with the NMR-derived restraints to model the 3D structure. And an example 3D structure you see here at the bottom, I have colored with dotted lines the NMR restraints we used to fold it. And in this slide, I will show you a cool video how our modified OpenFold predicts the structure of a rather large protein, 27 kilodalton. It's considered large for NMR. So with red dotted lines, you see the long-range restraints between distant residues. With green lines, you see the restraints between medium-range residues. And you can find the whole video on YouTube, on our YouTube channel. So usually after OpenFold, we do some equilibration in explicit solvent. And we do that using enhanced sampling molecular dynamics, particularly Hamiltonian replica exchange, along with NMR restraints, to produce the final ensemble, which you see at the right, because NMR is popular for providing, apart from 3D structure, also dynamics information, right? So everything will be available on our SaaS platform, which is currently under construction, will be available either online on the cloud or on premise. And these are the laboratories we have in Brno for protein production and biophysical protein ligand binding assays, like functional assays, for example. These are the NMR facilities we use. They are from our parent institute, SATEC. For NMR, we have all range of spectrometers up to 950 megahertz, which is suitable for the large proteins and the intrinsically disordered proteins. But we have access also to other biophysical facilities like microscale thermophoresis, SPR, mass spectrometry, which we use regularly for our drug discovery projects. In particular, we have 2 running drug discovery projects. One is on the androgen receptor. I already presented you the protein, and that is at the lead generation stage. And the second one we started this year is focused on an epigenetic target. The protein that is folded that you see here at the bottom, and it's at the hit discovery stage. And this is our current team. And we are looking for new members. So if you are compelled by the presentation, then please apply for these 2 positions. And I will be glad to answer any questions that you may have.
Thanks a lot, Thomas. So do we have any questions? And Thomas, I will ask you to repeat the question for the people online. So any questions for Thomas? Yeah. So if not, maybe I can ask you. So this.
But I understood anything.
So can you explain a bit more this concept of the ligand epitope?
Yes.
How do you actually calculate the?
All right. So I cannot disclose much information because have you noticed?
Yeah.
Thank you. Yes. So how we compute the confidence of the predicted ligand epitope, right? Was that the question? Good. So as you have seen here, this is under, we're filing a patent now, so I cannot disclose much information about the methodology. So essentially, we process these spectra, the one you see at the bottom, from the position of the quenching, we call it, the differences. We can predict which atoms, which protons of the small molecule interact. And these are subsequently provided as input to our neural network. It's tailored, made, and along with the chemical structures. So it can associate patterns, it can identify patterns of ligand epitopes occurring in new unseen molecules. And it can also give you some probability of whether these patterns are true. And that's how we quantify the predicted interacting atoms that you see in this slide. This one, for example, has a larger circle. It means that it's more likely to be the one that interacts with the protein. The whole molecule has been predicted to be a binder. That's essential. And the other one that have smaller circles means that it's less likely to be the main anchor points for binding, right? And what is important about NMR is that it can capture very weak interactions in the millimolar range, right? Unlike X-ray crystallography, for example. So for weak binders at the early stage of drug discovery, like hit compounds, which you cannot crystallize, then you don't have many options to study the interactions, in atomic level, I mean. So NMR is the primary source of this information. That's why we believe it's so important and it's unique, I mean, as you see. Thanks.
One more question over there.
Yeah.
So I asked this in the Discord, but maybe in here, you do structure predictions by IDPs. That's NMR that says the question I would have is, once you use the classical source of information about IDPs, how would you validate or structure predictions?
Okay, I'm going to repeat the question. He said that we do structure prediction of IDPs. I never mentioned that. And how do we validate that the structures we predict are correct? So exactly for the same reason that IDPs do not have a structure. Like here, you see, it's a chaos. I said that doing structure-based drug design here is futile, but what you can do is you can study the atomic interactions in the spectra. And that we achieve through the two-dimensional spectrum, like the one you see here. It's a two-dimensional. It gives you the atoms of the IDP that interact with the ligand. I mean, we're not, I'll say, delusional to try and model the 3D structure of these interactions because they're not stable 3D structures. But in the spectra, always tell you the truth. Yeah. Models don't tell you the truth. So we use the truth, the true information, to train the neural network instead of modeling something that is fictional and train the neural network with that. Did I answer your question? Partly.
Looking at how to validate beyond the
yes, we use orthogonal biophysical techniques like microscale thermophoresis. We prefer that because it doesn't require attachment to a membrane, like SPR, for example. So that's the method of choice. And of course, the one-dimensional NMR that we use from the perspective of the ligand. So these are the three primarily experimental biophysical binding assays that we use. One-dimensional NMR from the perspective of the ligand, two-dimensional from the perspective of the protein, and microscale thermophoresis.
So, thanks a lot. Okay.
Thank you.